Howdy, time for science. We're looking at amino acids and proteins. Let's have a look at the science understandings. Proteins are polymers of amino acids. Amino acids contain a carboxyl group and amino group. Amino acids are acidic and basic. They can undergo condensation reactions to make proteins. Amino acids, amide groups in proteins are peptide links or peptide groups. The structural arrangement of a protein depends on secondary interactions, and the biological function of a protein is a consequence of the spatial arrangement. So why are amino acids important? They're the building blocks of proteins. All your proteins are made up of a combination of 20 different amino acids in a particular order, and that produces all the different proteins in your body. So the structure of amino acid, there's an amino group, there's a variable side chain, and then there's a carboxylic acid group. This variable side chain, this is where the secondary bonding comes in that leads to different shapes of proteins. So what makes it an amino acid? There's a carboxylic acid group, that's the acid bit, and there's an amine group, amino bit. So amino acids have a carboxylic acid group and an amine group. So let's have a look at a couple of examples. Here's alanine. Now these are in what's called Zwitter-ion form. We'll talk about what that means in a second. But here's your amino group, here's your carboxyl group. And in this case, the variable side chain is just a methyl side chain. So that's a very simple amino acid. If we look at tyrosine, we can see that's a lot different. Again, we've got our amino group, we've got our carboxylic acid group, but in this case we've got a benzene ring, and that's an alcohol group at the end. These will have different attractions for different parts of the proteins. This is non-polar, this is quite polar. There is a non-polar aspect to this as well. So we'll get different secondary interactions. We'll talk about that a bit later. So here are the 20 amino acids that are in your body, and you can see all the variable side chains that are present. Some are very simple, some are quite complex, and again, we're going to be talking about the polarity of these side chains and how that affects the shape later on. So amino acids can self-ionize when you put them in solution. What that means is the hydrogen hydrogen ion proton from the carboxylic acid group can protonate the amino group to form an ammonium group. So we get this charge on here. That produces what's known as a Zwitter-ion. So this is the Zwitter-ion form of an amino acid. So we've taken the proton from the carboxylic acid group and we've protonated the amino group. Let's have a look at some example Zwitter-ions. So here's cysteine, and we can see we've got the basic amino group and the acidic carboxylic acid group. The hydrogen ions gone from the carboxylic acid group over to the amino group, and we've protonated that. It's the same down here for leucine, we can see here. So let's talk about proteins. Proteins are made of amino acids joined together, and you get lots of amino acids joined together and you make these very complicated structures. Different proteins have different jobs, and we'll talk about some of those jobs a bit later on. So proteins are made up of amino acids, and amino acids are bonded together with what we call peptide groups, but it's exactly the same as an amide group. It's just when it's present in a protein we call it a peptide group. So because of that, we call proteins polypeptides. They're polymers of peptide groups. So let's look at an example. So here we have a polymer strain. It's polymer because we've got lots of monomers joining together. In this case, we're joining each of the monomers together with an amide group. But because this is present in a protein, we call that a peptide group or a peptide link. So just restating that. So in proteins, the amide group that links the amino acid monomers together, we call that a peptide link or a peptide bond. So down here we can see here they come in. Okay, so we can see the C double bond ONH, C double bond ONH. That's joining our three amino acids together here, so we call that a peptide link or a peptide bond. So you may be asked to draw the structure of the monomers in a small polypeptide chain. So what you need to do there is you need to break up the peptide bond that's linking your amino acid monomers together. So the first thing you need to do is identify the peptide bonds, so that's the C double bond ONH that we talked about just before. Then you need to separate the monomers at the peptide bond by breaking between the C and the N. So here I'm drawing boxes indicating three monomers, and we're breaking the bond between the C and the N, because that's where the amide bond is produced. So then you draw the structures, and you need to finish off the groups that have been cut up. So in this case, you need to have a carboxylic acid group present, so you need to have C double bond ONH. For the middle one, you need to regenerate the hydrogen, and on the other side also the carboxylic acid group, and in this final one over here you just need to regenerate the amino acid group. Whenever you're doing that, you're putting in water to do this, so this is a hydrolysis reaction if it was a reaction. So we just separated a polypeptide into its monomers. When you join the amino acid monomers together, it's a condensation reaction because water comes out. So down here we can see we've got three amino acids that we're bonding together to make our polypeptide chain. you see that whenever we join it, we're reacting a carboxylic acid with an amine, so making an amide bond, and just with any other amide bond production, it's a condensation reaction, so water comes out. So let's talk a little bit more about proteins now. So proteins have different shapes, and the shape of the protein determines what it does. So we can see there's lots of different shapes of proteins, there's lots of different sizes, and it's the sequence of amino acids and the polarity of the amino acids, as well as other structures that are formed, that determines the shape. So here's some examples of some different proteins. We can see this, in this case we can't see every single atom that's involved in the protein, but we can see these whorls structures, so these whorls here, and these pleats here. They represent particular repeats of amino acids that lead to particular shapes. If we have a look over here, we can see we've got two proteins, actin and myosin. So actin's on top and in the middle here, and then we've got the myosin in the middle. These proteins, the shape of these proteins means that the actin can be ratcheted over by the myosin, so the myosin kind of pulls its way along the actin. These fibers join up together to make muscle fibers, so this is how your muscles work. And it works because of the shape of the actin and the myosin and how they interlink with each other. So you can get hydrogen bonding in proteins between sections of chains and between the chain and water. So let's have a look at that. So here we have a polypeptide. We can see we've got the variable R group. We see we get hydrogen bonding between the amino end, the amide group, and the carboxyl end of another one. So we can get hydrogen bonding between those. You'd also get hydrogen bonding between the amino acids and water molecules nearby if it's in solution. So here again we've got a carboxyl, the carboxyl end, the acetyl bond O part, carbonyl, that should be, sorry. Carbonyl end, the oxygen is delta negative, so that will attract the delta positive hydrogens from the water molecule. Then we've got the amino end as well, so we've got the NH here, so the hydrogen is delta positive and will attract the delta negative oxygen from the water molecule. So you get hydrogen bonding between the proteins and water, as well as sections of the chains as well. Now we're going to quickly look at proteins as enzymes. We've looked at this before, but we'll just go into a bit more detail here. So biological enzymes are catalysts. They speed up chemical reactions. The way they do this is reliant on the shape of the enzyme. So let's have a look at that. So here we have the substrate. This is the substance that we're going to change, and here we have our enzyme. The substrate makes its way into the active site on the enzyme. When that happens, we get attraction between parts of the active site and the substrate, and that can cause a change in shape of the enzyme, which can break bonds or force bonds to form between parts of the substrate. In this case, we're breaking the substrate in two. So the enzyme stretches or flexes and breaks some bonds, and then you get your product being produced. So this provides the orientation part of the requirement for a successful collision between reactants. So the reactants are in the right orientation, the bonds are broken, so it requires a lot less energy to do it rather than just colliding the particles together successfully. So let's have a look at some active sites. So here's an enzyme. Here we can see this empty space in here. That's the active site. And here we can see the substrate going into the active site. When that happens, the enzyme changes shape slightly. It flexes and stretches bonds in the substrate, and we can lead to your product being produced. Let's have a look at some examples. Over here we have lactase metalloproteinase. We can see the active site in here. There's several copper ions in there. This could be used to break down estrogens in wastewater, and we're going to look at wastewater treatment a bit later on. In wastewater recycling, one of the problems is estrogens and phytoestrogens and synthetic estrogens going in the water supply as a result of people taking the pill. And you need to remove those estrogens from the water, otherwise that can lead to effects if you recycle that water and people drink it. So there's a possibility that this enzyme could be used to break down those estrogens, and that would make the drinking water safer to use. Over here we can just see another protein, and again we can see the where the active sites are on this protein. You can see the molecules that are interacting there at those active sites. So now we're going to have a look at the effect of pH and temperature on proteins. We can talk about this in terms of enzymes, but the enzymes in your body will only work over a certain temperature range and also a certain pH range. If the pH or temperature changes too much, then you change the shape of the protein, and the shape of the protein is fundamental to what its usefulness is. So let's have a look at how that happens. So let's have a look at the effect of pH changes first. So pH changes affect the bonding between ammonia side chains and carboxylic acid side chains. So these can be attached to several amino acids. If you reduce the pH, so that means you're making the solution more acidic, the carboxylate anion groups can be protonated again, and that reduces the strength of the negative attraction down here, and that reduces its polarity. So here we have a full negative charge. When we put the hydrogen on here, we don't have that full negative charge anymore. It's still polar, but it's not as polar as a full negative charge would be. Similarly, if we increase the pH, we're making the solution more basic. So if we take, say, a protonated amino side group, it will lose the extra hydrogen ion if we put that in a basic solution because you're reacting a base with an acid. This is acidic here because we've got the extra proton there. So what that means is you regenerate the amino group, and again, that reduces the attraction between, so if this is positively charged, it would attract, say, a negatively charged carboxylate anion group of another part of the protein. If you get rid of that charge, the extra positive charge there, then you're going to reduce the attraction here, so the protein might not stick together as much. So both of those changes, by either adding acid or base to proteins, can lead to the side groups changing their structure, which reduces or increases their polarity, which will lead to a difference in shape. So let's have a look at the effect of temperature now. So any temperature above 50 degrees will break most of the secondary interactions that are occurring between the protein chains. And those are things like hydrogen bonding and dipole-dipole, as well as dispersion forces. When you're heating up these proteins, you're giving them extra energy essentially, and that's going to make the proteins move more, and as they move, they'll get further and further apart. The different side chains will get further and further apart, and they will break the hydrogen bonds or dipole-dipole interactions or even dispersion forces. So what can happen is the protein will unravel into an unusable shape. So down here we have the functional protein, but we heat it up and we get this structure that's all stretched out, and we would call that denatured. So today on Flipping Science, we looked at amino acids and proteins, and why proteins are important. That's it for Flipping Science today. See ya.